Cerebral is a tool for systems biologists to visualize and explore quantitative data from multiple experimental conditions. The data is overlaid on a biomolecular interaction graph, laid out using annotations about the location of the biomolecule in the cell. Cerebral is implemented as a plugin for Cytoscape, a tool which loads biomolecular interaction graph models and associated node and edge data from multiple sources. Our plugin replaces the standard graph viewer. Many layouts optimize graph topology but ignore biological metadata. Here, the colors show the membrane-bound compartment where a biomolecule is located in a living cell. Molecules from different compartments are mixed together in the graph view. In contrast, Cerebral divides the nodes into layers according to compartment in a familiar cross-sectional view of the cell common in cellular biology diagrams. For immunology data, proteins activated in response to infection are shown in a special layer below the nucleus, clustered together into groups with shared biological function. Biologists can position nodes by hand and pin them to form a skeleton of important biomolecules. The biologist views experimental data by selecting node attributes corresponding to measurements collected for each node in the graph. Each experimental condition is mapped to a small multiple view and an axis in the parallel coordinates view. The values associated with a node map to an intersection point on the axis and a color coding in the small multiples view. The size and number of columns in the small multiples view is adjustable. The user can reorder conditions by dragging axes or the small multiple views. The reordering is linked between views. We reorder the values to place the time series in order and each control LPS condition beside the treatment LL37 condition. Looking at the small multiples, the LL37 treatment has the largest effect at hour 4. We compute a direct calculated difference in the main view and see that most genes are colored orange, indicating repression, particularly amongst the inflammation-causing cytokines and chemokines. We can examine the same data set in the much larger MAP kinase interaction graph. Here we use mouse hover highlighting and linked panning and zooming to explore a more complex interaction model. Turning to a second example, we investigate the Spellman yeast cell cycle data. We have expression levels measured at 24 time points. Selecting individual time points color codes the main window accordingly. We use the parallel coordinates display and clustering to investigate the data. Exploring different k-values, we notice that some clusters exhibit a sinusoidal pattern. Clicking the cluster selects the corresponding nodes. We see that 6 of the nodes are highly connected in the graph. Selecting just these 6 nodes shows even tighter clustering in the parallel coordinates display. In fact, these are the histone genes, and biologists will recognize that their expression level follows the cell cycle changes. This is visible both in the parallel coordinates view and in the small multiples view.